My name is Slavica Tudzarova. I am Associate Professor at the Division of Endocrinology at the David Geffen School of Medicine. My background is in pharmacology and tumor biology. I received my PhD at the Medical University in Vienna, Austria, before joining University College London to answer the fundamental question how metabolism is linked to the cellular decision to replicate, let's say to regenerate and forge tissue regeneration. At that time, I worked with Sir Salvador Moncada, multiple Nobel Prize nominee, now my lifetime mentor and advisor to Metanoia Bio. Metanoia Bio is the preclinical startup based on patented and licensed technology from UCLA, where I'm the main inventor. So the main discovery is that the tissue show imminent property to recognize injured cells based on relative fitness differences between healthy functional cells and those which are old, senescent, damaged, etc. So they are able to replace those damaged cells, which triggers functional cell regeneration. Now, the problem occurs within the chronic damage, such as, for example, diabetes or neurodegeneration, where those chronically damaged cells actually change their behavior by switching to a different way they produce energy, which is oxygen independent, and with that they achieve to survive in the face of the injury, but at the same time to disguise themselves so they cannot be recognized and eliminated by healthy cells, which basically impedes functional cell regeneration. What we learned is how to deprive those cells from their survival mechanism so that we reveal their true identity of damaged cells. They can be recognized by healthy functional cells, so unleash this functional cell regeneration. So Metanoia Bio is linked, the co-founding of Metanoia Bio is linked to a little bit of anecdote. Namely, in the time of the pandemic, by the force majeure of being locked or under lockdown, we were confined to the residential lobby as our remote working place with the chairman of Metanoia Bio, Mr. Brent Cohen. And so inevitably, we were exposed and overhearing our own stories and the theme of our work. He became truly inspired by the excellent scientific work which was ongoing at UCLA. And that is how it started, the exposure and perpetual introduction to his both business and clinical network, based on which Metanoia Bio was co-founded. Metanoia Bio is currently at the stage for ask of 6 million funding to support its MedChem program that's supposed to generate oral, patentable, novel, new composition entities, so clinical candidates that will induce functional beta cell regeneration in type 1, type 2 diabetes, and diabetic retinopathy. And although this path is not easy, it's often deemed as difficult, tenacious, and laborious. At the same time, we are very inspired because we are in front of 500 million people to which we can possibly offer a solution.